Hello and welcome to our webinar on porcine CD4 T cell immunology. I am Oliver Rosenwasser and I am the product manager for the veterinary antibody products at ABD Serotech. I will introduce you to our speaker in a moment, but first would like to let you know that this webinar will be recorded and an on-demand version will be available soon on the ABD Serotech website. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up the technical guide, which gives solutions to the most common technical problems. We would like to make this webinar as interactive as possible, so please submit questions or comments via the Q&A box located at the bottom of the screen throughout the webinar. We will answer as many questions as possible at the end of the presentation. Should any questions remain unanswered, we will email you a reply. And I would like to introduce Professor Armin Saalmüller. Professor Saalmüller holds the chair of the Institute of Immunology and is head of the Department of Pathology at the University of Veterinary Medicine, Vienna, in Austria. His group's research is focused on porcine T cells and cellular immune responses against viral infections. Current research priorities are the detection of these immune responses and the identification of immune cells and their functional characterization to gain a better understanding of the interaction of the immune system with pathogens. In collaboration with leading vaccine manufacturers, the ultimate goal is to contribute to the development of new or improved vaccines and to improve diagnostic tests. And now, over to you, Armin.
So hello everybody. Thank you for joining my webinar. It's a pleasure for me to present you in the next 45 minutes some data of our work. So I want to talk about the role of CD4-positive T cells in an antigen-specific immune response, about CD4-defined T cell subsets, about regulatory T cells in swine, about IL-17A-producing T cells, the CD8 alpha expression on porcine CD4-positive T cells, about porcine TH1 cells, multifunctional T cells, the expression of CD27, the differentiation between central and effector memory cells, and the contribution of NK cells and gamma delta T cells to the polarizing TH1 milieu. This slide gives you a simple overview about the immune reaction against vaccines or pathogens, starting from interferon alpha producing plasmacytoid dendritic cells, Interferon-Gamma producing NK cells. We have, on the other hand, immature dendritic cells which maturate to conventional dendritic cells, presenting foreign antigens in the lymph node to CD8-positive T cells or CD4-positive T cells. Some of the CD4-positive T cells help B cells for differentiation into antibody-producing plasma cells and memory T cells. And on the other side, we have in the T cells also effector cells, memory cells which can differentiate into central memory and effector memory cells. In this presentation, I want to focus on the CD4-positive T cells with their different functional subsets. There's a current paradigm of T helper cell differentiation. We start from the CD4-positive T lymphocyte, and this T lymphocyte maturates under the polarizing milieu into the respective T cell subsets with a different functional activity. We have the cytolytic T lymphocytes on the right. We have the TH1 cells characterized by TBIT expression. We have follicular T helper cells. We have TH2 cells characterized by GATA3 expression. We have TH17 cells characterized by the transcription factor Rho gamma T. And we have the regulatory T cells characterized by FOXP3 and expression. And these cells maturate to different effector cells. They produce different effector mediators, and they have different functions. Killing of infected cells by the cytolytic CD4-positive cells, macrophage activation by the TH1 cells, germinal center help by the follicular T helper cells, immune response against helminths and allergies by the TH2 cells, inflammation by the TH17 cells, and we have a regulation and suppression of the immune response by the regulatory T cells. The big question at the moment is, what is known for swine? So in this presentation, I do not want to talk about CD4-positive cytolytic T cells, which play a role after viral infections in swine. I also do not want to talk about follicular T helper cells, and I do not want to mention the GATA3 expressing CD4-positive TH2 cells. The main focus on my talk will be on TB-positive TH1 cells, raw gamma T TH17 cells, and FOXP3-positive regulatory T cells. I want to start with the regulatory T cells stimulated by the polarizing milieu by IL-2 and TGF-beta, with effector mechanisms IL-10, cytokine, and with their effector function, regulation, suppression of inflammatory responses. The porcine regulatory T cells are defined by CD4 and CD25 antigen expression, and in addition by intracellular expression of FOXP3. When we have a look on the CD4 T cell subsets, within the CD25 negative ones, we can't find any FOXP3 expression. Within the CD25 dim ones, we have a small population, a mixed population. Some of them express FOXP3, and within the CD25 high positive T lymphocytes, most of them show FOXP3 expression. For evaluation of the suppressive activity of the CD4, CD25 defined T cell subsets, we stimulated PBMC with anti-CD3 antibodies and measured the 3H-thymidine incorporation after 4 days cultivation. You see the positive effect of CD4 positive, CD25 negative cells, and you see the inhibitory effect of the CD4 positive, CD25 dim population and the CD4 positive, CD25 high population on the proliferation of our PBMCs. Regulatory T cells are defined by their IL-10 production. And when we stimulate sorted CD4, CD25 defined T cell subsets with CON-A and measure their IL-10 production over the time, you see the CD4 plus CD25 dim T cell subsets represents the most IL-10 producers. The CD25 high CD4 positive T cells show minor IL-10 production. To get an impression of the targets of suppression of the porcine Tregs, we stained PBMCs with CFSE and added CD4, CD25 defined T cell subsets or potential regulators to these responders. We stimulated the cells with CON-A and looked for the proliferation over a period of 4 days. When we have a look on PBMCs on day 4, you see quite a nice proliferation without this potential regulators. you get a negative suppression by adding the CD4 plus CD25 minus subset. We get a intermediate suppression by adding the CD4 plus CD25 dim positive ones, and we get a clear suppression when we add CD4 plus CD25 high positive ones. And we have a look on the targets of suppression. You can see that T helper cells, CD4 positive T helper cells, CD8 beta positive cytolytic T lymphocytes, as well as gamma delta T lymphocytes are suppressed by our CD4 plus CD25 high positive regulators. So I come now to the first summary. You can say that porcine regulatory T cells, or Tregs, express FOXP3. Most of porcine Tregs belong to the CD4 plus CD25 positive T cell subset. And within the subset, two major groups can be discriminated. We have the CD4 plus CD25 high positive cells, mostly FOXP3 positive, which show suppressor activity. And we have the CD4 positive CD25 dim cell subsets, which contains mainly IL-10 producers. We can say that porcine Tregs suppress the proliferation of various T cell subpopulations. I come now to the next T cell subset, to the TH17 cells, and here again the question, what is known for swine? In swine we can show that CD4 positive T cells as well as gamma delta T cells show an IL-17A staining. And in regard to the CD4 positive T cells, there's a big question. Which CD4 positive subset contains the IL-17A producer? For swine, it is known that besides the CD4 positive, single positive, and the CD8 positive cytolytic T cell subsets, an additional subset of subpopulation of CD4 positive cells exist, the CD4 plus CD8 alpha positive T lymphocytes. So when we have a look on the phenotypic changes during porcine T helper cell differentiation, we can say that resting T helper cells express CD4, but they are negative for CD8 alpha, they are positive for CD45RC, and they are negative for MHC class II. After stimulation, the cells acquire CD8 alpha, they downregulate CD45RC, and they also upregulate MHC class II expression. So we have activated T helper cells being CD4 plus, CD8 alpha plus, as well as memory T helper cells, which also show CD8 alpha expression, downregulation of CD45RC, and also they are also positive for MHC class II. And when we have now a look on the phenotype of our IL-17A producing T helper cells, we see that all co-express CD8 alpha and belongs to the CD4 positive, CD8 alpha positive T lymphocyte subpopulation. So in regard to differentiation of TH17 cells, we can say that CD4 single positive cells are stimulated, and under the influence of IL-6 and TGF-beta, they developed to CD4 plus CD8 alpha positive TH17 cells producing IL-17. And in parallel, there's a stimulation of gamma delta T cells, and maybe this gamma delta T cells show a synergistic effect in regard to IL-17 production and in regard to inflammation. To the next summary, you can say TCR gamma delta and CD4 plus CD8 alpha CD4-positive T cells produce IL-17A. The CD4-positive, CD8 alpha-positive T cell subpopulation includes activated T helper cells and memory T cells. But there are still some open questions. What is the role of the CD4 plus CD8 alpha-positive T cells and the gamma delta T cells in inflammation processes in swine? What's the role of the IL-17A producing T cells in the immune response after vaccinations? Do they play a role in bacterial infections? We do not know at the moment. We come now to the TH1 cells. I think the best characterized T helper cells in swine. They are polarized by IL-12 and Interferon-Gamma, and they are characterized as main Interferon-Gamma TNF-alpha producer. So in the next few slides, I want to focus onto the TH1 cells characterized by the expression of Tbet and the production of Interferon-Gamma and TNF-alpha. So when you look now for the antigen-specific Interferon-Gamma producing cells, we use ELISpot assays. And here, 4 samples are presented. We have the immune response of Interferon-Gamma producing cells against PERS virus, the upper line, in comparison to mock control. We have the immune response against flu A, in comparison to the mock control, against Salmonella typhimurium and against M. hyo. And you see this especially in PERS infected or PERS vaccinated animals, a high frequency of Interferon-Gamma producing cells. The same do we have in flu A virus infected animals. We can now compare the number of the Interferon-Gamma producing cells stimulated with the respective antigens in comparison to mock and medium, and PERS compared to flu A showed the same amount of Interferon-Gamma producing cells in 10 to the 5th ppmcs. The next big question is, we have the same amount of antigen-specific cells. The big question is the frequency of the Interferon-Gamma producing cells an appropriate parameter for the quantification of the cellular immune response? And second, what do we know about the quality of the Interferon-Gamma producing cells? When we are talking about the quality of an immune response, multifunctional T cells are mentioned as a potential correlate of protection. What are multifunctional T cells? Multifunctional T cells are single T cells which produce simultaneously several effector molecules, like cytokines, for example Interferon-Gamma, IL-2 and TNF-alpha, chemokines, and are molecules involved in degranulation. And we were now looking for multifunctional T cells in swine producing Interferon-Gamma, IL-2 and TNF-alpha. For the identification of multifunctional T cells in swine, we used monoclonal staining with monoclonal antibodies against Interferon-Gamma, TNF-alpha and IL-2. And you see here the expression of the respective cytokines in CD4-positive T lymphocytes. This is in flu A, after flu A restimulation. We get nearly the same picture when we restimulate CD4-positive T cells with PERS virus. We get also an Interferon-Gamma, TNF-alpha, and IL-2 production. And for a better characterization of this multifunctional T cells, simultaneously producing Interferon-Gamma, TNF-alpha, and IL-2, we used Boolean gating, and we found, in regard to influenza restimulation, a lot of triple producers, some Interferon-Gamma single producers, TNF-alpha single producers, and interferon, quite a nice amount of Interferon-Gamma and IL-2 double producers as well as interferon-gamma TNF-alpha double producers. We could now compare the reactivity of our cytokine-producing T cells in regard to influenza, reactivity against influenza virus and reactivity against PERS virus, and we saw differences in the amount of triple producers and double producers. But to date, we do not have an explanation for these differences, and this is part of our current work at the moment. I come now to the third summary. I think we could show that interferon-gamma ELISpots are a robust and sensitive method to identify the involvement of T cells in the immune response towards vaccination or infection. But a correlation towards protective immunity may not always exist. We favorize multifunctional T cells to play an important role in protective immunity. This identification of multifunctional T cells is possible in swine. Their role as reliable correlates of protection needs further investigation. And there's now the question, what is the phenotype of the interferon-gamma producing T cells? When we stimulate lymphocytes with PMI ionomycin and look for their interferon-gamma production, you can see that mostly CD4-positive T cells produce interferon-gamma. And when we further look for the phenotype of these interferon-gamma producing cells, most of them are CD45RC negative, and half of them are either CD27 positive or CD27 negative. What is now the role of CD27? What is now the role of CD27? In our hands, CD27 is a novel marker for detailed discrimination of CD4-positive T cells. When we have a look on the CD4-positive T cell subpopulations, the CD4-positive, CD8 negative ones and the double positive ones, we see that all naive CD4-positive, CD8 negative ones show a high CD27 expression, whereas the double positive ones, they split into two CD27 defined subpopulations. We can find the same pattern in spleen and the same pattern in lymph node. But in lymph node, the CD27 positive ones are enhanced in the CD4 plus CD8 alpha positive subpopulation. Therefore, we set up following hypothesis. We have our CD4 positive, CD8 alpha positive activated T helper cells, and when they further differentiate, they become CD27 positive central memory cells and CD27 negative effector memory cells. To get an impression about the function of the CD8 alpha, CD27 defined CD4 positive T cell subsets, we sorted the cells into three different fractions, into CD8 alpha negative CD27 positive naive T cells, into CD alpha positive CD27 positive cells, and into CD8 alpha positive CD27 negative cell populations. So in the first approach, we studied the cytokine production of the CD27 defined T cell subsets and could show that the CD8 alpha, CD positive, CD27 negative ones were the main interferon-gamma producers, as well as also the TNF alpha producers. In regard to IL-2, the naive CD27 positive, CD8 alpha negative cells showed the highest reactivity. When we stimulated our 3 CD8 alpha CD27 defined T cell subsets with ConA, we could show that the naive in blue, they showed a nice proliferation after stimulation. The CD27 positive CD8 alpha positive ones showed similar proliferative capacity, whereas the CD27 negative ones, they showed only, if at all, an abortive proliferative response. In regard to other differentiation antigens, we studied the surface expression of CCR7 and the messenger RNA of CD62L and CX3CR1. In regard to CCR7 expression, we could show that our CD27 negative subset within the PBMCs represented 2 subpopulations, whereas in spleen and lymph node we could find only 1 subpopulation. CD27 CD8 alpha positive ones are represented in PBMC, spleen, and lymph node, similar way. And in regard to CD62L expression, it's downregulated on CD27 negative ones, whereas CX3CR1 expression is upregulated on the CD27 negative ones. This data confirm now our hypothesis. We have central memory cells showing CD27 expression, CCR7, as well as CD62 expression and CX3CR1 expression. And on the other hand, we have effector memory cells which downregulated CD27, shows an intermediate CCR7 expression, downregulated CD62L, and upregulated CX3CR1. Come now to the next summary. In swine, we have central memory T cells with the phenotype CD4 plus, CD8 alpha plus, CD27 plus. And we have the highly differentiated effector memory T cells with the phenotype CD4 plus, CD8 alpha plus, and CD27 minus. These cells produce high levels of interferon-gamma and TNF-alpha. They show a reduced proliferative capacity. They are mostly CCR7 minus and have downregulated CD62L. And they show an upregulation of CX3CR1. So we have now clear data about these 3 CD4 defined T cell populations in swine, about the regulatory T cells, about the TH17 cells, and about the TH1 cells. And in regard to the TH1 cells, there's a big question. Who is responsible for the polarizing milieu, for the production of IL-12 and for interferon-gamma? When we have a look on the differentiation of the porcine TH1 cells, we start with the immature dendritic cells, maturated to the conventional dendritic cells, have the interaction of dendritic cells with the CD4-positive T cells, and the big question is now, who is producing the interferon-gamma necessary for the polarizing milieu? That the CD4-positive, CD8 alpha-positive TH1 cells develop and produces interferon-gamma and TNF-alpha. Appropriate candidates are natural killer cells. They can be stimulated by IL-12 and IL-18 produced by the conventional dendritic cells, as well as IL-2, and we know that they produce interferon-gamma. So when we look on the phenotype of the porcine NK cells, we can say that these cells are CD3 negative and most of them are CD8 alpha positive. And within PBMC, we can distribute these NK cells to two different subsets, to NKp46 negative ones and to NKp46 positive ones. And when we look in spleen or other lymphoid organs, we found in addition another subpopulation showing a downregulation of CD8 alpha and a high NKp46 expression. When we looked for the function of these three NK cell subsets, we could show that the NKp46 high subset are the main interferon-gamma producer. But there might be an additional source of interferon-gamma, and this additional source are gamma delta T cells. These gamma delta T cells, they can be stimulated by IL-12 and IL-18, and they produce, in addition to the NK cells, interferon-gamma. So what is known about porcine gamma delta T cells? These cells are CD3 positive, and they are characterized by the expression of gamma delta T cell receptors, in this case stained by an antibody against delta chain. And this is a population with two distinct subsets. Both are CD4 negative. Some of the gamma delta T cells show no expression of CD8 alpha, and some of them show a clear CD8 alpha expression. And as indicated, this TCR gamma delta T cell subpopulation is not a homogeneous population. It's somehow quite heterogeneous, as presented here by the expression of CD2 and SWC5. SWC5 means swine workshop cluster 5 monoclonal antibody. It's an undefined antigen with a molecular mass of about 100 K. By staining of CD2 and SWC5, we can define three subsets: the CD2 positive ones, the SWC5 positive ones, and the double negative gamma delta T cells. And when we look for the interferon-gamma production, we can say that the main interferon-gamma producer are the CD2 positive SWC5 negative ones, here presented in red. They produce interferon-gamma after IL-2, 12, and 18 stimulation, as well as additional stimulation with concanavalin A. So when we look now to the differentiation of the porcine TH1 cells and the cytokine milieu and the respective populations producing the cytokines, we can say that interferon-gamma is produced by NK cells, stimulated by IL-12 and IL-18, as well as gamma delta T cells, which produce also a huge amount of interferon-gamma and contributing therefore for the TH1 polarizing milieu. So, I think we can summarize this data again. The polarization into TH1 phenotype is driven by interferon-gamma. NK cells are an important source for interferon-gamma. And the CD8 alpha low positive NKp40 high positive NK cells show the highest interferon-gamma production. And these cells represent a highly active NK cell subset. Another statement is that porcine T cells contain a substantial amount of TCR gamma delta T cells. And the CD2 positive gamma delta T cells are the main interferon-gamma producers. Together with NK cells, TCR gamma delta T cells contribute to the polarization of the CD4 positive T cells and the generation of an Interferon-Gamma TH1 milieu. I come now to the final summary and some additional thoughts. I think we can say that CD4-positive T cells play a central role in the modulation of the immune responses of the vaccination. And differentiated CD4-positive show heterogeneous functions. The question is which CD4-positive subsets should be involved in protective immune responses. TH1 cells, TH17 TH2 T, FH, I think rather than regulatory T cells. But we have also to differentiate between pathogens. We can say that the differentiation of naive CD4-positive cells is driven by the polarizing cytokine milieu. And this polarizing cytokine milieu is generated by antigen-presenting cells, by NK cells and gamma delta T cells. The question is now, how can we steer the differentiation of appropriate CD4-positive subsets? I think we need more defined adjuvants for a more targeted stimulation of cells of the innate immune system. There's an additional question of shall we stimulate NK cells and gamma delta T cells for a better immune response? And what ligands can be used for that? I think I've shown that multifunctional T cells producing interferon-gamma, TNF-alpha, and IL-2 can be identified in swine. But their possible role as correlates of protection needs further investigation. An additional point is that porcine interferon-gamma producing TH1 cells can be discriminated by their CD27 expression in tricentral and effector memory cells. There's a big question, can the generation of respective antigen-specific memory T cell subsets serve as indicator of an effective immune response? We do not know at the moment. But I can say the first steps are made, but there's still a lot to do to understand the immune response of swine after infection and after vaccination. At the end, I want to say thank you to all of my co-workers at the Institute of Immunology at the University of Veterinary Medicine in Vienna, Austria, as well as to our partners at the University Clinic for Swine, as well to our sponsors, the government of Niederösterreich, the FFG, COMET program, the Ministry of Science, and our industrial collaboration, IDT and Boehringer Ingelheim.
Thank you, Armin, for your interesting and very informative presentation. For our audience, please enter your questions now in the Q&A box at the bottom of the screen if you haven't already done so. Before we move on to the Q&A section, I would like to briefly highlight a few of the key antibodies that Serotek has to offer to support research into the porcine immune system. As we have heard today, measuring and understanding immune response is a very important requirement to support the development of new vaccines. Serotek manufactures antibodies that will enable you to detect the major cell population in pigs in flow cytometry experiments. Among them, T, B, NK, other cells from the innate immune system, and endothelial cells. Here on this slide, you can find a few of the antibodies available from Serotek to define these populations in swine. Please find these and more at serotek.com/vet. And now, let's open the Q&A session. Okay.
Hello.
So, thank you for your questions that have appeared. We will pick about 4 or 5 questions, and then any remaining ones we can answer on email. So, Armin, for the first question, Are the CD8 alpha molecules expressed on CD4-positive T cells monomers or homodimers?
As we know, Oliver, the CD8 alpha molecules are homodimers composed of two CD8 alpha chains.
Uh-huh.
This is
Good.
different from the CD4 negative, CD8 alpha positive cytolytic cells. These cells show CD8 alpha, CD8 beta heterodimers.
Okay. The next one. Do TH2 cells exist in swine?
Yes, they exist, and they were characterized, for example, in Ascaris zoom-infected swine by an increased GATA3 expression.
Okay, thank you. Is porcine CD27 expressed on B cells?
No, CD27 in swine is not expressed on cells. This is in contrast to human and mouse.
Aha, that's where that question came from. Good. Do we have an explanation for the different phenotypes and functional distribution of the TCR gamma delta T cells?
There are several hypotheses. One can speculate that these phenotypically different TCR gamma T cells belong to different T cell lineages, I would say. But we have shown manuscript published about 1.5 years ago that we are able to generate in vitro, at least in vitro, CD2-positive gamma delta T cells from CD2 negative. This might give a hint for different maturation stages of gamma delta.
Okay. And finally, what is SWC5?
SWC5 describes the swine workshop cluster 5, which was introduced in 1992 on the first international swine cluster of differentiation workshop in Budapest in Hungary. SWC describes molecules with no known counterparts or autologs in the human CD nomenclature. But in the meantime, some of these molecules have been better characterized by molecular approaches. For example, SWC1 is now CD52. Our SWC2, which we defined in Budapest, is CD27. And SWC3, a marker for myeloid cells, is CD172A. SWC5 seems to be autolog, the bovine WC1, a member of the scavenger receptors, cysteine-rich family, CD163. But this has to be shown on molecular level. That's future work.
Uh-huh. Okay. Well, it now just remains for me to thank you, Armin, for your time, and thank our audience for showing up and for supplying some excellent questions. There are some more questions there. We will get to those by email. And thank you very much, everyone, again. And thank you again, Armin, for a very interesting and very informative talk.
Thank you very much, Oliver, for the invitation. And thank you to all visitors and listeners for your time. Have a nice evening or have a nice day. Goodbye.
Okay. Goodbye to everybody.